Hello, everyone. Welcome to today's live broadcast, an unbiased view of the next generation sequencing market. We are delighted to bring you this educational web seminar presented by LabRoots. I'm Judy O'Rourke, and I'll be your moderator for today's event. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. I would now like to introduce today's speaker, Shawn Baker, co-founder and chief science officer of AllSeq. I will now turn it over to Shawn for his presentation.
Thank you for the introduction, Judy, and thank you all for attending this presentation. So as was stated, this will be an unbiased view of the next generation sequencing market. And what I want to do is take you through a few of the trends that we've seen at AllSeq. But before I do that, I need to explain to you who AllSeq is and why we think we have this unbiased view. AllSeq has created the sequencing marketplace. So this is sort of like Match.com, but rather than being for people looking for dates, it's for researchers who are looking for sequencing services. So what we've done is we've built a series of free online tools that helps researchers describe the experiments that they want to do, their sequencing experiments, and then we match them up with the providers who can offer those services. But a key point of this is that we don't actually offer the services ourselves, and we don't own any sequencing instruments. So we feel this gives us an unbiased, neutral standpoint. And when you combine that with a large number of experiments that we see coming through the sequencing marketplace, we feel it gives us a really unique perspective on the market, really interesting insights. And it's some of those insights that I'm going to share with you today. Okay, so let's start in with the first trend. This is the big one that everyone's been talking about for a little while, and that's the $1,000 genome. And the big thing here is that it's finally here. This is something that's been talked about for a long time, so we're all really excited that we finally have it, sort of. And there's some caveats here that I'm going to walk you through. So the first thing is, what was able to generate this $1,000 genome? And that's the HiSeq X Ten instrument from Illumina. So this is their latest and greatest machine. It is capable of generating 600 gigabases of output per day. So that translates to over 1.8 trillion bases per run. And that's enough data for that instrument to generate around 5 whole human genomes per day. And that translates to over 1,800 genomes per year when you run it day in and day out, which is what they expect. So how does this work? What's it got that's different from the machines that used to be out there? Primarily, it's these pattern flow cells. This allows Illumina to pack in a lot more data per run and a lot easier prep for it as well. And of course, when you have more data at higher density, you need improved optics to be able to capture all that data. So that's basically what they've done. So this sounds really great. Latest and greatest from Illumina, the market leaders, lots and lots of data. What's the catch here? The catch is that while there are lots and lots of things that people like to do with their sequencers, lots of organisms they like to sequence and lots of applications they like to run, the HiSeq X Ten is highly limited. In fact, it can only work with one organism, and that's humans. And not only that, it can only do human whole genome sequencing. So that means that you can't do other really interesting things like exome or targeted sequencing. You can't do RNA-Seq, can't do all sorts of things, just human whole genome sequencing. But if you can live with that limitation, it's actually a really nice low-cost output system. So the main goal here is to get that $1,000 genome. How exactly does that happen? How does that break out? That $1,000 breaks out like this. About $800 of that goes to the actual sequencing reagents, the juice. $135 goes towards paying off the instrument or the amortization. That leaves about $65 for everything else, which is primarily library prep. That includes not only the kits and the reagents to run the library prep, but also the labor. So obviously you've got to be a pretty efficient factory to get your costs down that low. It also, this $1,000 doesn't include everything. There's things like overhead, the building that the instrument is housed in, the electricity to run it, turning on the lights. It doesn't include data storage or any kind of involved data analysis. But it does do a pretty good job of delineating what is available so people can calculate their actual costs. All right, so what would this mean if you wanted to get your own $1,000 genome? What would you have to do? First thing you got to do is you've got to buy the instrument. And these are pretty pricey instruments. They cost $1 million, and they're the most expensive next-gen sequencers on the market today. But the thing is, you actually can't really just buy one. It's called the HiSeq X Ten, and that's because you've got to buy them in multiples of at least 10. So $10 million is your minimum input just to get started. But once you've done that, you're off to the races. You're sitting there generating some data. So how much is it going to cost you to actually do this? Well, I talked about how there's enough data generated on these instruments for 5 human whole genomes per day at $1,000 each. That means every day you have this instrument turned on, it costs you $5,000. And of course, you need to be running this day in, day out. So over a year, operating that instrument is going to cost you $1.8 million. And again, this is the HiSeq X Ten, so you had to buy 10 of them. So it's actually going to cost you $18 million a year to operate these instruments. And to get that instrument amortization down to only $135 per genome, Illumina had to stretch it out to a 4-year lifetime. So that means over 4 years, you're actually going to have to spend $72 million to get that $1,000 genome. Now, all is not lost. AllSeq is here to the rescue. Illumina has actually sold about 20 of these instruments and groups of instruments to researchers and groups all over the world, and we have several of them that are offering services on the on AllSeq. So you're more than welcome to use us to try to get access to these instruments. Okay, the number 2 trend that has made a lot of progress over the past year is that of long reads. So Illumina is the dominant player here, but they're really known for their short reads. But long reads are starting to come into fruition. So why do that? The short reads have actually been really, really successful. Lots of great research, lots of great papers have come out about it. But why might you want to actually use longer reads? One of the primary reasons for doing that is to look at haplotyping or phasing. So here I'm showing you just one stylized example of a gene with 3 SNPs. We've got an A to T change and then 2 C to G changes. And so if you were to sequence this with a short read sequencer, that's what you would see. You'd see the normal sequence, and then you'd see a mix of the altered sequence, or these SNPs. But the thing is, for diploid organisms like humans, this is actually a mixture of 2 different alleles, one from the mother and one from the father. And if the reads are short enough that they don't span multiple SNPs, it's actually really difficult to tell what the actual allele structure is. And in this one particular example, we've got a couple options. So it could be that these 3 SNPs are actually distributed across the 2 alleles so that you've actually got defects in both copies. Or it could be that all 3 defects are in one allele, leaving the other allele completely intact. And this, of course, can have some really significant consequences in medical genetics, whether or not you have an intact copy of the allele or not. It can be the difference between having a disease and being completely phenotypically normal. So obviously a really important application. Another reason that long reads are desired is getting a good understanding of what the actual repeat structure is. So here I'm showing an example of a CGG trinucleotide repeat. And this is a repeat that is present in humans and other organisms, and it can be related to developmental delays. So you can have anywhere from just maybe a couple dozen of these repeats all the way up to a few hundred repeats. And if you have just short reads that don't span outside the smallest region of this repeat, you actually can't really map this with any precision. You don't know what the underlying structure is. So you won't be able to tell the difference between what would be the normal condition or the condition that would confer disease. However, if you have long reads that span through the entirety of that repeat structure, you can uniquely map them, and so you can get a lot more information here that just simply isn't available with the short reads. Another example, kind of similar thing, we're looking at some structural variations here. So on the top, we have just a simple repeat. On the bottom, we have an inversion structure. And again, if you have short reads that don't span outside this repeated region, it'll map equally well to all possibilities, so you won't be able to tell what's going on. Again, if you have those long reads, they'll be long enough, they'll span outside the repeat region into the constant region, and they'll be able to be completely unambiguously mapped. So again, revealing a lot more information that you just really couldn't get from the short reads. Sometimes there's some tricks you can do, but in general, there's some very serious hard limitations on what you can do with those short reads. So obviously long reads then have a place, and the leader in generating those long reads is Pacific Biosciences. So they've been out for a couple of years and have been making some really strong improvements in this area. Their average read length is now up to about 15 kilobases, and that compares to roughly 200 or 300 base lengths from the other technologies. So we're talking almost 2 orders of magnitude increase in read length, and they're improving all the time. Now, there's a few negatives with this technology. One, the instrument's quite expensive. It's a $700,000 instrument. It's also very heavy. It requires specialized flooring to install it. The other thing is kind of an interesting plus and minus. It has a really high error rate. So an error rate is hovering around 13%, and that compares to around 1% down to even 0.1% for the other leading systems. So substantially higher error. But where it's actually a slight positive is in that the error model is stochastic or random. So what this means is if you take the same region of the genome and sequence it multiple times, you're very unlikely to see the same error more than once. So what that means is if you sequence deeply enough, you can actually get really high quality, really low error rate consensus sequence, and actually even better than the lower error rate models from Illumina, because those actually have some systematic error that no matter how deeply you sequence, you're always going to have the same problem over and over again. So for really high quality sequence, PacBio actually is a good choice. It's just more expensive per output and for the instrument. So another technology that's used for generating long reads that people are getting quite excited about is Oxford Nanopore's MinION. And as the name would imply, this is a nanopore-based sequencer. And there's a lot of really interesting, exciting things about this. And the primary one is the fact that it appears to be a nanopore sequencer that actually works. So nanopores are a technology that people have been looking to and trying to develop for at least 20 years and continually talking about the promise of it. But until recently, you couldn't actually do it yourself. But now the MinION has been created. It's actually a really small sequencer. That's that little box right there in front of the laptop computer. It's a little handheld USB-driven sequencer. It can currently generate around a gigabase per day, but the more important thing is that the read lengths are very long. So just like PacBio, it's got read lengths that can easily go past 10 kb. There have even been some hints that it can go as high as 100 kb or so. Kind of really depends on how intact your DNA is. Now, it does have some issues. Just like PacBio, the error rate is quite high. So the people who've been using it are reporting error rates anywhere from 10 to 30%. So that's extremely high. The good news, though, is that it is improving. So it's going through rapid iterations of the chemistry, and that's really helping with the error rate. So in-house, Oxford says that they've got that error rate down to around 5%, definitely under 10%. The other thing about this that's kind of unusual is that it isn't really a commercially launched system. So it's a little bit difficult to know exactly what's going on. The good news is it is available in an early access program, and the other good news is that early access program is really quite widely distributed. So there's several hundred different research groups all over the world who are using this instrument. But it still does make it a little bit difficult to know all the details. Because it's early access, they don't have an actual pricing model. They haven't really talked about exactly what that's going to cost. The other thing is it's still a little bit early on, so with that high error rate, we don't know exactly what the error model is. It's looking pretty good, like it can be used to create very high quality sequence, but it's still not clear if it really has that true stochastic model that PacBio has. So it's something we're going to keep an eye on. In fact, they just had a meeting today called London Calling, where they talked about a number of improvements. It just happened, so I'm still trying to digest it all. I can't share it all with you, but it looks like they've done a number of nice things to it that are going to come out over the next several months to a year. Things like faster throughput, so it'll really increase the throughput and lower the cost. Changing some of the physical components to it to lower the cost as well, and also increase the number of cores. So all this should translate to a machine that is easier to acquire and produces a lot more data. But we're just going to have to see exactly where that pans out. They also talked about the next instrument. This is the PromethION. This is basically a series of those MinIONs put all together. So it's a higher throughput machine. They talked about some of the specs here today. Again, it's a bit early, but once they have all the components rolled out, they claim to be able to produce lots and lots of data with this, terabases of data, just like that HiSeq X Ten, but presumably in a faster time frame and for less money. Now, this one isn't yet available in early access, unless they announce that today or maybe tomorrow. I'm expecting to see some news there. But for right now, it's something that we're just going to kind of keep an eye on, and it's definitely an interesting thing to look forward to see what Oxford Nanopore does. So these two examples that I showed you were actual true long reads, but there's another strategy you can take, and that's to create synthetic long reads by sort of piecing together a bunch of small reads. And one of the first companies to do this was Moleculo. So this is a company that was acquired by Illumina a couple of years ago, and Illumina then incorporated it into their product lineup. They changed the name to TrueSeq Synthetic Long Reads. It's a bit of a mouthful, so a lot of people just still call it Moleculo. So what Moleculo does is they generate roughly 10 kilobase synthetic reads. So the way they do this is they take an entire genome and then they chop it up into roughly 10 kilobase fragments, and they distribute that across a 384-well plate. Inside each individual well, that DNA is further fragmented and a unique barcode is added. Then the DNA in all the wells can be pooled back together. It can be additionally fragmented, amplified, and then sequenced just using the normal Illumina machines. The way that it becomes interesting is that because those unique barcodes were added, the DNA from each well can actually be bioinformatically separated from all the other wells. And so what that means is when you go to assemble this and align it to the genome, instead of trying to do that with the entire genome at once, you can actually treat each well as a little mini assembly project. And the result of each little mini assembly project is a small number of those 10 kb synthetic reads, giving you essentially 10 kb of read. It's not quite as good as a true 10 kb read. It's got some limitations or some repeat structures that it can't really work through, but it does give you a nice way of giving more information than you could get with just the short reads themselves. There's another company called 10x Genomics that just came out of stealth mode a couple months ago. They're doing something similar, creating these synthetic reads, but they use a different process. So rather than using a 3D4-well plate, they're actually looking at beads in an oil emulsion. So obviously, we're talking much smaller volumes. 10x also uses a much larger number of barcodes, so they can deal with many more beads. Moleculo is talking about 384 wells. We're now talking about maybe hundreds of thousands of individually addressed beads. So lots more barcodes, lots more runway. Also, because of the way they amplify the DNA, they can get longer reads. So the Moleculo method uses some form of long-range PCR, which kind of tops out at around 10 kb. 10x Genomics is doing something else. It allows them to amplify much larger fragments, so they think they can get synthetic reads that are about 100 kb. And then the final difference is that 10x is an automated system. So this has some pluses and minuses. The minus is it's a machine you have to buy. So that's about a $70,000 instrument. Pretty small though, desktop machine. The good thing about it is that it's automated, so it's actually much easier to use. So people are expecting this to be an easier system for people to adopt in the lab. Okay, so that was trend number 2 of long reads. The third trend I want to talk to you about is clinical sequencing, and this one obviously has a lot of importance for how sequencing is actually being used. Up to now, up to at least the past year or so, next-gen sequencing was really geared just towards researchers. They're the ones that all the companies were trying to sell to. And so therefore, they geared their machines and their kits and their whole way of thinking to what researchers want. What do they want? Researchers want increased output, they want lower cost of that output, and they want rapid updates. As soon as anything new is available, they want to see it, they want to have it in their lab, in their hands. Clinicians, on the other hand, have a completely different perspective. What they want to see is they want a machine that's extremely easy to use. They want a really quick turnaround time. They're dealing with a patient sample, so they need to have a result as quickly as possible. And perhaps most importantly, they want that machine and that system and that set of reagents and even the analysis of it to be as stable as possible. So while researchers would love to have ease of use, and they would certainly love to have quick turnaround time, this stability is directly opposed to the idea of rapid updates. So this is a major change that the sequencing providers, the platform providers, have had to come to terms with. Basically, those clinicians have to give up those rapid updates in order to get the stability. And if a researcher wants to use a clinically valid system, they have to give up the idea of getting those rapid updates. So question that we get a lot is, this is all great, but when will next-gen sequencing actually be adopted in the clinic? The answer is it already is. So one of the first applications to really take off was non-invasive prenatal testing, or NIPT. It's a way of testing fetal DNA from a simple blood draw from the mother. And this one is really taken off and shown to be a pretty big market and a growing market. Another one, of course, is cancer diagnostics, and then more recently, a subset of that is liquid biopsy. So again, kind of like NIPT, rather than biopsying the actual solid tumor, looking for clues about the tumor in the blood. So basically, tumors, as they metastasize and as the cells die, they shed off some DNA into the blood, and at high resolution, that can be detected with these next-gen sequencing systems. Another one is the use of NGS in rare childhood diseases. So this is probably not a big market, but it's an important one. Children who have developmental problems, who've gone through all the other diagnostic processes and not had any answers as to what's going on, those patients are often referred to next-gen sequencing to try to figure out, is there something else in the genome that they can use to try to, at the best, give them a treatment, at least just understand what's going on and maybe avoid some painful treatments or some expensive treatments. And then the last thing that we're starting to see is the conversion of single gene genetic tests into these multi-gene genetic tests. This would have a couple of advantages, primarily probably in terms of ease of use. So rather than doing serial tests one after the other to try to figure out what's going on, running them either all at once or many of them at once, that can speed things up and also reduce cost. So we're already seeing this in the market today, and we expect to see this just expand over time. So there are a couple platforms that are on the market that are specifically cleared for clinical use. The first was launched by Illumina, and that's the MiSeq DX. This was launched kind of end of 2013. It was supposed to have a follow-on with the HiSeq, which was going to be released for the NIPT test, but it turns out once Illumina made their sort of mid-range machine, the NextSeq, they decided that was a better fit. That's probably the next FDA-cleared instrument we're going to see from Illumina. Illumina has been working with a number of pharmaceutical companies to generate universal NGS oncology tests and oncology companion diagnostics. So that's a way to get the technology basically adopted in the clinic as quickly as possible, getting those pharma companies on board. Illumina's competitor, Ion Torrent, which is owned by Thermo, followed on about a year later with their clinically cleared instrument. That's the PGM-DX. And again, just like Illumina, they came to the same conclusion that getting pharmaceutical companies involved, creating these universal NGS oncology tests, are really a good way of helping getting these technologies adopted. So there's a couple solutions already available, and more are likely to be on the way. A big thing for clinical adoption is also going to be interpretation. So harking back to this example I showed before, looking at this one stylized gene, the clinicians don't want to see this. They don't want to see the individual sequence. They don't even want to really see the individual SNPs. They want that turned into something they can use, a true interpretation report. They want something that will tell them maybe not exactly what to do with their patient, but give them the tools they need to figure out what they should do. What drugs should they take? What treatment should they have? What's the likely prognosis? And so really getting to that interpretation report as opposed to raw data is going to be critical. So naturally, there's a lot of companies who are interested in helping them do this. I've just shown you a few examples here on the screen. This is a very dynamic field. We see a lot of these companies pop into existence and pop out of existence every year. And it's one of the areas where we really kind of expect to see some consolidation, which brings us to the fourth trend that we see in this market today, and that's consolidation. So the first company to really do this in a big way is QIAGEN. They already had a strong hold on the sample prep market, but they wanted to have tools that would take the user through the entire workflow, from sample prep, sequencing the data, to then analyzing the data. So the first thing they did is they went and bought a sequencing company. They bought Intelligent Biosystems, or IBS. This is a company that's using a similar kind of technology to Illumina, sequencing by synthesis. It supposedly has some advantages, lower cost, lower error rate, and the instrument they're working on is supposedly going to be pretty different. Rather than building a big one that can handle a lot of samples at a time, they're looking to create a relatively small one with a modular structure that can handle one patient sample at a time. Unfortunately, we haven't seen too much about this. They bought this company, I think, over 2 years ago, and they still haven't launched it. We don't have a hard launch date, but hopefully that's progressing. We don't really know. But QIAGEN has purchased some other companies as well to fill out this workflow. They bought CLC Bio, which helps them with the raw analysis of the data. They bought Ingenuity, which helps with interpretation. And then they bought Biobase, which has some biological databases which that interpretation engine can use. So if they get that sequencer to work, they will have a pretty nicely filled out workflow here. But we're still waiting to see all the pieces that finally come together. Another company that's made a lot of moves in this area is Roche. So a couple of years ago, Roche, who was actually the first next-gen sequencer on the market, they came out with a 454. They realized that their machines were getting outpaced by Illumina, so they tried to buy Illumina. Fortunately or unfortunately, depending on your perspective, Illumina didn't want to be purchased, so they spurned that advancement. And after a while, Roche figured it had to give up. So they stopped trying to acquire Illumina. They also did some other things. They announced the end of life of the 454, so it's actually going to no longer be supported after mid-2016. So that's basically a dead man walking kind of platform. They also killed off a number of the partnerships that they had working with research groups and other companies to try to come up with sort of the next generation of sequencing that would be even better. And they killed off a bunch of them. So it really seemed to a lot of people, myself included, that it just looked like they had given up and they were going to just get out of the next-gen sequencing space. But it turns out that's not what they did. They just decided to restart and retrench, and they've made some fairly bold moves in terms of acquisitions and putting some money into some other companies. So again, they wanted to fill out this workflow covering sequencing, analysis of the data, and then actual applications of using all that together. So they acquired Genia. Genia is another Nanopore company, somewhat similar to Oxford Nanopore. It uses a slightly different technology. And we don't actually know too much about it these days because they went kind of dark after Roche acquired them. Roche also made an investment in Pacific Biosciences. So they tasked them to take their RS2, their big, giant, long read sequencer, and make a desktop version of it that would be useful for the clinic. And that apparently is progressing. We don't really know the specifics of it. It's a private collaboration, and then they're not really telling the progress, but we do know that Roche has been making milestone payments for that. So clearly PacBio is making progress. They also invested in a company called Stratos. Stratos Genomics isn't a sequencer per se, but what they do is they're creating some technology that can help the detection of DNA, especially for nanopore systems. So while they haven't said it explicitly, it certainly looks suspicious like maybe Genie will start using Stratos technology in the future, assuming things go well. Roche also purchased Bina, which creates data analysis, online solutions, and hardware that can accelerate the whole process. So they're trying to get that down. And then they also bought a couple companies, Ariosa, which is an NIPT company, and Cap Medical, which is not too much is known about them, but they're looking at cancer liquid biopsies. So this is taking that next-gen sequencing technology and actually applying it. They've also made a major investment in Foundation Medicine. They are probably the #1 cancer diagnostic company using next-gen sequencing, and Roche now has a majority stakeholder there. So they're making some pretty bold moves as well, really trying to bring next-gen sequencing to the market and also kind of leapfrog the technology that's out there, trying to come up with something better. So we'll see where that goes. Okay, so those are the 4 major trends that we see. Another thing that I wanted to share with you today is some of that unique information that we have access to that other people don't. So we took a look in aggregate at all of the projects that were going through our sequencing marketplace and pulled out some data that we think is kind of interesting, and that's what I want to share with you. So probably the easiest thing to look at is platform preference. I think if you did a poll, everyone would get this question right. What's the #1 platform? It's Illumina. There's really no question about that. So for people using the sequencing marketplace who had a preference, around 83% of them wanted an Illumina system. So while we would have guessed it would have been #1, I'm not sure I would have guessed that it would have been so high. The other interesting thing is that for second place, we actually have a tie between Ion Torrent and PacBio. So in the past, Ion Torrent has really been kind of the #2, the main competitor to Illumina, and PacBio is more of a niche player. But Ion Torrent, unfortunately, has been pretty delayed in their latest product releases, and PacBio has been making some steady progress. So if that continues, we would actually expect PacBio to take over as the #2 position. And then you have the older systems of 454 and Sol that kind of round out the rest, but those are really dying off pretty rapidly. In terms of applications, again, maybe not a huge surprise here. DNA applications account for about half of all the projects that come through our sequencing marketplace. And this is roughly split in equal portions of whole genome sequencing, exome sequencing, and then other forms of smaller targeted sequencing. The #2 application then would be RNA, or transcriptomics. And the vast majority of this is what I would call true RNA-Seq, looking at the full transcriptome. There's a little bit of small RNA projects and just a little bit of that older tag-based DGE type method, but the vast majority is the RNA-Seq. And then there are a bunch of much smaller, more niche type applications, ChIP-seq, metagenomics, methylation, things like that. So they're still there, people are using them, but they're much smaller proportion compared to the bulk, which is DNA and RNA. In terms of species, this is another one that was maybe a little bit surprising to me. You know, if you read the trade journals and you look at the popular press, pretty much all you hear about is humans. You know, how is this going to be used for health? What is this going to mean for you directly? And it is true. It is the #1 species that we see coming through the sequencing marketplace, but it's actually less than a third. That means there's a whole lot more variety out there. Basically, you know, bacteria, plants, mouse, fish, other mammals, just all sorts of things. Anything that can be sequenced basically is being sequenced. A couple of the interesting species we've seen come through, there's the Gayal, which is some sort of African bovine species. And then another one, this kind of creeps my wife out, it's the deep sea spider. So basically just a tremendous variety of stuff coming through. Humans are important, but over 2/3 are other organisms. So this last one is a really important question. We get it asked all the time. what does sequencing really cost? And so you can look at list prices, you can try to see what people are saying, but when it comes down to it, actually having contracts saying you want X, you'll pay Y, we actually have that data, so we know exactly what these sequencing projects are costing. So probably the one that people care about the most, human whole genome sequencing, kind of a standard 30X project, the so-called $1,000 genome. Big surprise, it costs more than $1,000. Of course, there was the cost I said weren't included in the $1,000, things like the overhead, the data storage, data analysis, but also in this case, profit. These are service providers who are offering this, so they need to pay their clientele, or rather their personnel, and they need to make a profit. So the average price for this is around $1,775, a little under $1,800, and there's some variability here. That's a standard deviation of a little over $200. So what causes that variability? Really, the number one thing is number of samples per project. So if you come to a provider with just a handful, 1, 2, maybe 5 samples, you're going to pay a higher price. But if you come to them with much more, 50, 100, even 1,000, you'll start seeing some serious discounts, and you can get prices quite low. We even see prices as low as that sort of magical $1,000. It's not common, but we have seen prices that low. Other things that can impact the cost is the starting material. So obviously, if you bring a pre-prepared library, that's going to cost less than just raw DNA. And then the data analysis as well. If you're willing to take back raw DNA, that's the cheapest. Standard data analysis alignment's pretty basic, not too expensive, but then if you want something more, that's going to cost you more. Another major application is human exome sequencing, and this is at a standard is at 100x. This one is interesting because it's being used for a variety of reasons, but one of those reasons is that it's been substantially cheaper than whole genome sequencing. And it's still true that it is cheaper, but it's not as, the difference in price isn't as great as it used to be. And that's because the HiSeq X Ten really brought the prices down, but only for whole genome sequencing and not for exome sequencing. So as we see here, the average exome project price per sample is a couple $100 less than whole genome at $1,500, but it's got a huge variation. That standard deviation is almost $1,000. And there's some things that impact that price. Just like whole genome, the number of samples per project, obviously the more samples you have, cheaper it's going to be per sample. But the much bigger thing seems to actually be the data analysis. So with whole genome sequencing on the X10, more people seem to be willing to just get the data back. It's being used more for just research projects. Exome projects are skewing much more toward clinical and actual patient samples. And so data analysis becomes much more important. They want that additional analysis added on, the interpretation of the genome. And that's really going to increase the price quite a bit. So that's why the price difference between whole genome and exome isn't quite as high as you might have otherwise expected. So the final kind of major application I want to talk to you about is RNA-Seq. And I've normalized this to 10 million reads. That's kind of on the low end in terms of what people typically would run. This would give you roughly equivalent results to a microarray. And it's the lowest price project type that, at least compared to the DNA ones, at a little under $600 per sample. But as you can see, that standard deviation is nearly 500. So a huge amount of variation here. Again, kind of broken record, samples per project. The bigger the project you have, the cheaper it's going to be per sample. But a much bigger thing that was impacting price in the project that we saw was read length. So if you go with a very simple 1x50, single read, 50 base read, that's going to be on the cheaper end. If you want something more involved, looking at splice junctions and things like that, you might want that paired end 200 base reads. That's going to be a lot more expensive. So that's a huge factor in determining the end cost of these kinds of projects. The other thing that impacted the price pretty greatly that was a bit of a surprise was the species. So if you're doing something that's been well studied, looking at human, mouse, yeast, things like that, it's pretty cheap to get analyzed. But if you're doing something that's not so well studied, maybe doesn't have a good reference genome, the price really goes through the roof. And that's probably because you're asking that provider to run de novo transcriptome analysis every time. So there's a lot more labor, a lot more artwork involved in that, and the price really goes quite high. So there's so much variation here that I actually wanted to show you some of the data, kind of how it spreads out in a histogram. You can see over on the left that the bulk of the projects are in the $200 to $300 or $400 range. That's pretty close to probably what people are paying for a microarray through a core lab or through a service provider. So that's pretty much parity. But you can see on the right-hand side, things can get very expensive, over $1,600, $1,700 per sample. And that would be, again, for those specialty type of projects, maybe a species that hasn't really been sequenced before. So it becomes a true research project. Okay, so that's kind of what we're seeing today. There's a few things that we would like to keep an eye on in the future, and we think you might like to do so as well. Obviously, we expect some improvements from Illumina. So changes to the X10, maybe eventually being able to buy just one of those instruments, an X1. They actually have already released something called the X5. So if you don't have the $10 million to buy 10 of these, they will now sell you 5 of them for $6 million. So you can save yourself a bit of money that way. Unfortunately, they then jack up the reagent cost when you do that. So the cost for generating whole genome with the X5 system is $1,400. But still, it gives people a way to sort of buy their way in. The other thing that we're anticipating potentially happening is getting more applications to open up on the X10. As I said, right now it's only human whole genome. The obvious thing to do would be to open it up for exome sequencing. It turns out there's a hint that maybe it's actually already been opened up for whole genome bisulfite sequencing. There's one of the service providers out there tweeted out that they're doing this, offering this as a service, and Illumina retweeted it. So they clearly know about it and are happy with it. I don't know any of the details, but it is an interesting clue that at some point that machine is going to start opening up to other applications. We're also keeping an eye on those new platforms. I already mentioned that PromethION system. I'm not quite sure when that's going to come out, but if it's able to do all the things that Oxford Nanopore says it can do, that's going to be a real game changer for people. Another system is the Complete Genomics system from BGI. So Complete Genomics is a company that BGI bought a couple of years ago, and up until recently, it was operated strictly as a service. So you would just send samples to Complete and BGI, and they would process it on their little factory floor and send you back the data. But it turns out they're actually turning that into an instrument that they can ship to people, and they have hinted at it, and later this summer they're going to hopefully tell us more information. Basically, they've hinted that it's going to be a lower cost system, definitely competing with the X10 and having high quality data. So definitely something to keep an eye on. A couple of the systems that we're interested in but haven't heard too much about is Genapsys and Genia. Genapsys uses a technology that's somewhat similar to Ion Torrent, but they claim through their different chemistry to be able to get longer reads and higher quality reads. Genia is that Nanopore company I talked about before. They had been releasing updates to the public, letting people know what was going on, but since they were purchased by Roche, everything kind of went dark. The good news, though, is that Roche appears to be putting money and resources into that technology, so hopefully they're progressing, and hopefully, I'm not sure when, but hopefully at some point we'll start seeing that system out in the wild. The last thing really to keep an eye on would be data interpretation standards. Not just the analysis and the interpretation. This is especially important on the clinical side. It's always a balance of trying to decide when those standards need to go to be implemented. If you bring them on too early, you can actually limit the advancement. But if you wait too long, especially things like clinical adoption can really be slowed down. Physicians who are using this, they don't want to have to guess at which way to align their data, which way to do the SNP calling. But they want some standards in place that they know they can rely on and they can trust. And what they do today is going to happen same tomorrow, next week, next month, next year. So that's something we're looking forward to as well. So that's what I have for you today. I appreciate everyone taking time out. I would also, there's some Q&A that's going on here, so if you have any questions you haven't thought of, please put them in there. I would also encourage you to check out our website, follow us on Twitter, and if you have any other comments or questions that you didn't think of today, please feel free to email me directly. Thanks a lot.
Thank you, Shawn, for that informative presentation. Before we get started on the question and answer session, I'd like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. And our first question is, can all sequencing applications be utilized on the MinION?
Yeah, that's a great question. Can all the applications be utilized in the MinION? I would say not yet. It's still in this early access program, so we've seen people do a lot of interesting things with it. It's obviously being geared more towards small genomes because the output is still relatively small. You know, in the best cases today, people are getting about a gigabase, but a lot of more typical use might be more like half a gig or so. So it's somewhat limited there. I would say that things like RNA-Seq will be a challenge for a little while until they get the throughput up, just because you tend to need a large number of reads. That said, at their London calling meeting, apparently they talked about doing RNA sequencing. You know, they've talked about a number of things. Really, what's missing so far from that system is the upfront sample prep. And they've kind of talked about not needing sample prep. They've hinted, not more than hinted, they've talked about this new instrument whose name I can't remember. They just talked about it today. That's going to automate some of that sample prep. So I think once that gets out there, then we'll start seeing a lot of other applications. There's no obvious limits to what that machine should be able to do. The only exception I could think of would maybe be small RNA. It's not that they couldn't do it, but that's probably not going to be the strength of what that Nanopore system can do, which is really good at looking at long reads. Obviously, with small reads, you're not going to get that advantage. So thanks. Great question.
How does one utilize the early access program?
The early access program for the MinION is what I'm assuming. That's relatively easy. You would just apply for it. You know, I don't know the details, but if you go to Oxford Nanopore's website, they are definitely taking people in for that, and it does not appear to be particularly exclusive. So, you know, the more traditional early access program is really meant to be looking, or rather working with a small number of researchers who are pretty friendly to the company who's doing it. It's highly controlled. There's a little bit of that going on, but this isn't your typical early access program. There are literally hundreds and hundreds of independent researchers who are doing this. So, you know, if you have trouble finding it yourself, just email me and I'll help you find the link. But basically, it's just going onto the link, filling out the application, deciding how many you want access to at a time. The last I looked, it was basically, it cost you a $1,000 refundable fee per flow cell that you want to work with at a time. So if you want 5 at a time, that's $5,000. Once you use them, I think you maybe ship the used ones back and get new ones shipped back to you, and then just at that point you're paying shipping costs. So it's not particularly difficult to get in, but you do have to apply. Next question.
Regarding cancer-targeted gene sets, what is popular in your view? What are the best options?
So targeted cancer gene sets. You know, I can't give you a list of specific genes. There are a few solutions out there. Basically, what people seem to be focusing on is roughly sets of up to 200 to 300 genes. I think that Foundation Medicine has roughly 300 genes now, and so they're using a specialized version of targeted sequencing. So one of the issues that you have with targeted sequencing is it doesn't always pull down all the regions equally well. And so what they've done, and probably what some other competitors have done, is they've taken time to kind of tweak that pull down process to make sure that they get uniform coverage, because that is fairly critical. In terms of which ones are best, I honestly can't tell you. I can say that they're pretty much all using the Illumina technology, so it's high quality sequence with some known limitations, but they would sort of design around those limitations. And then it's maybe not very satisfying, but I guess the only thing I can leave with you is it's clearly that Foundation Medicine is the leader in that area, and you know, they just had a huge influx of cash from Roche, so they clearly thought Foundation has something that maybe is a cut above the rest. Hopefully that answers your question.
Do you think that the next-gen sequencer producing companies will be willing to invest in developing sequencers for short reads around 1 kb for the projects that only require them instead of long read for lower prices, or will that portion of the market be forced into using those next-gen sequencers in the future?
So I'm not completely sure I understand the question, but I'll answer it as best I can. And then what I would say is if I didn't answer what you were really looking for, just email me and I'll try to do a better job in person. So what we're seeing right now and what's completely dominating the market, just completely dominating it, are the short reads. And that's for a couple reasons. The cost of those reads on a per base or per gigabase metric is far and away the lowest. So the lowest cost you can get there with a HiSeq X Ten is about $7 per gigabase, and that's just so much lower than all the other technologies. You know, haven't really priced it out exactly, but just as a comparison, the PacBio machine, which is producing the longest reads, it's able to generate about a gigabase, but closer to $500 or $1,000. So it's substantially more expensive per base to do it that way. So because of that reason, short reads are dominating. Another reason that short reads are good is if you can run a lot of them. So it's kind of naturally, it's easier to run a lot of short reads than running a lot of long reads in general. That said, if these other technologies come out and they continue to improve on generating the long reads, they bring the cost down per base, and if they increase the throughput, so you're getting lots and lots of reads, they will start to look a lot more attractive. There generally aren't any truly inherent advantages to the short reads. It was just easier to do. So in terms of what the market's going to look like, it is difficult to say, but my guess and my hope is that we will see different types of sequencers serving different types of markets. And the truth is that that's true today. It's just that they're mostly coming from one company. So Illumina built the HiSeq X Ten. That's for you know, country-wide sequencing projects. You want to sequence everyone in a country or really, really large cohorts. And that's the cheapest per base. But then you've got, on the other end, you've got the MiSeq, which is much more expensive. It's closer to $100 per gigabase, but it's quicker, the machine's cheaper, the output is smaller, so it's really better geared towards small instruments. So I definitely think you're going to see different instruments geared for different types of applications. And, you know, Illumina is not the only company to do that. Ion Torrent has tried to do that a little bit with their two machines, the PGM and the Proton, and Oxford Nanopore is talking about that as well. The MinION is going to be the little handheld portable that can go out in the field. The PromethION is going to be that bigger instrument. Not so big, so maybe it'll go in the field as well, but that's going to be more the workhorse if someone wants to sequence a whole human genome or several whole human genomes. So yeah, I think you will see lots of different manifestations of these platforms serving the different market needs. And again, if I didn't answer that exactly the way you were really looking for, just please give me an email and I'll try to give you some more info. Great question.
Can you comment on the cost of RNA-Seq at 50M? Would that be 50,000 reads?
Yeah, so that's 50 million reads. And again, a good question. The data I showed was actually normalized to 10 million. Basically, 50 million reads, it's obviously it's going to be more expensive. It's not going to be quite 5 times as expensive, though. It's not quite linear. There are some upfront overhead things, sample prep and things like that. So, you know, I haven't looked at those specific prices, but I'm going to give a rough ballpark of being something closer to $1,000, roughly in that general area. I will say, and it's something that we actually encourage people to do, the best way to find out what the prices are is to actually ask the providers directly, and that's why we built the sequencing marketplace. It's a way for you to pretty easily get prices from multiple providers in a really competitive situation, so you can really get the actual price there. But as a ballpark, I'm going to guess, you know, maybe around $1,000.
Do you think that the cost for WGS will reach the $100 mark or lower in the next 5 to 10 years?
That's a really good question. The answer is I hope it will. It's 10 years is really hard. Even 5 years is quite hard. In the past, prices have certainly been dropping fast enough that would be hit no problem. But a couple things happened. One, the more easy gains had already been made, so the lower you go, the harder those gains are. Second is that the competition kind of faded away a little bit, and right now Illumina is a little bit too dominant. They really control prices in this market. So what we need are a couple things. We definitely need to see some more competition. So Oxford Nanopore is certainly saying they're going to offer that. Maybe BGI will come back with their complete genomics and offer competition. But the other thing to realize is that while the sequencing costs have really come down a whole lot, the sample prep costs haven't come down. So even if you got sequencing costs down to 100, you still might be paying $50 or $100 for the sample prep. So in order, at some point, if the sequencing comes down far enough, it's going to put pressure on other aspects of that whole workflow. The upfront sample prep, and then also the downstream data analysis and interpretation. They're going to become bigger components to that. But certainly, based on the things that we've seen companies claiming, there's no reason to think that it couldn't hit the $100 mark, as long as the market operates in a relatively typical fashion, meaning that there's competition and that the market space increases. And that's really why all of these sequencing companies, they want to get into the clinical market, because that's a much bigger market than the research market. So if that happens, and it happens in a way that's relatively free market oriented, then that, my guess is, certainly by 10, I would really hope we'd be getting down to the $100 range. And I really hope we do, because I think a lot of really interesting science and a lot of really interesting health monitoring is going to start happening when that sequencing gets that cheap. So yeah, great question.
Okay, we have time for one last question. Where do you see the clinical pharmacogenomics market in the near term and long term? Specifically, what about reimbursement for the testing services?
Yeah, that's a really good question, and it's a point that I actually bring up a lot. And what you're gonna find out is I probably don't have a very good answer for you, but you did bring up a really good point. People ask, you know, what's holding next-gen sequencing back from the clinic? You know, what does the technology need to be? What does it need to cost? What does the error rate need to be? You know, all this kind of stuff. But the truth is, the thing that's really going to hold it back is that reimbursement. And the truth is, I just don't know. My guess here, and this is really just my opinion, it's not particularly based on any kind of deep analysis. My guess is that it will be reimbursed, and if it's not reimbursed, I think there'll be such a demand from the end users or the patients that they'll go around the normal health system that we have today. So at least in the U.S., I know it's radically different in other countries. Here in the US, it matters, is that procedure covered by insurance? Is it reimbursable? And that can be really slow to have happen. And if it's too slow, I think that the patients will demand it, and then the providers will probably respond by having more rational pricing for people who are actually, paying their own money to get it. So it's a little bit tricky to say. I know people are working on this. There's a lot of companies who are working. There's new CPT codes that are out there that people can start using now to get next-gen sequencing services reimbursed. But my guess is that the technology is going to develop so much faster than the bureaucracy of both the FDA and of the health insurance companies that it might just kind of be taken over by consumer demand. So again, really great question. So I guess that's what we had time for. I appreciate everyone attending, and I appreciate all the really wonderful questions. You don't always get good questions, and you guys did a wonderful job. Again, I'd encourage you to check out our website, follow us on Twitter, and if I didn't get to your question or if you'd like a little follow-up, please just email me at shawn@allseq.com. Again, thanks so much for joining today.
Today's webcast will be available for on-demand viewing through November 14th, 2015. You'll receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Goodbye.